Hello, welcome to today's episode of the OUR Path to Research podcast. Today we will be talking about the world of material sciences and alternative proteins. Today we have our wonderful guests.
So my name is Rakesh Sahu, and I am an adjunct associate professor in the Department of Material Science and Engineering, and also an associate member of the School of Biomedical Engineering. I am the Director of Operations for a research center, the Center of Excellence in Protective Equipment and Materials, and also I manage a research program in the area of cultivated meat, which is the alternative protein.
That's very cool.
And again, my name is Daniel Sim. I am a PhD student with Dr. Sahu, and my focus on my project is using alternative proteins as scaffold systems for cell culture.
That's really cool. Thank you. So for the first question we have for Dr. Sahu, could you briefly tell us about your research and explain what you do?
Sure. So as I mentioned that I have two different hats in the university. The first is as the Director of Operations for a research center, the CPEM, Center of Excellence in Protective Equipment and Materials. So that is a new research center that started during the COVID days. And our mission is basically to support the R&D for the companies and industries in Ontario and within Canada for developing new materials in the area of PPE, which is personal protective equipment.
Mm-hmm.
Now, in the PPE space, the most commonly used materials are the nonwovens, which is essentially a textile but made with random fibers distributed and being bonded by some means.
Mm-hmm.
And at CPEM, we have a lot of different tools and manufacturing capabilities to make these nonwovens. Although our mission is to support the research and R&D for the companies, we also have in-house research in developing new formulation, new materials formulation for these nonwovens that will allow us to make new functional PPEs and as well as add some new functions and functionalities. On the other side, as I mentioned, that I'm also managing a research program which is federally funded, and this is the largest federal funded grant within Canada in the area of cultivated meat. And this is a multi-institutional grant, which means we have partners and researchers from different universities across the Canada. We have people from different institutions working in different areas in the area of cultivated meat. But at McMaster University, our group primarily focuses on developing scaffolds that would basically allow us to create different meat configuration, for example, say meat patty or a steak. So we could make those. So we are trying to build those technologies and processes using a different combination of tools and processes and to enable making or fabrication of those meat architecture. Additionally, we also work on increasing the efficiencies of bioreactors. What bioreactors are, basically, these are large vessels. Imagine the breweries tanks, right? Mm-hmm. So these are large reactors where the cells are grown at a massive scale. And what we're trying to do is we're trying to increase the efficiency by having a larger cell growth and also increase the media recycling in the process, because that is a component which is a bottleneck in case of upscaling of manufacturing of this cultivated meat. Mm-hmm. So we are trying to increase the efficiency by combinatorial approaches. One is to increase the efficiency by reducing the shear stress, improving the cell health, and second is to increase the recycling of the media so that we can reuse the same media for a longer duration of time, and we don't have to replace this media quite often as that is traditionally used.
It seems like you're addressing so many different challenges, so cool. Whether that be in health, whether that be in food and nutrition, and then cell culture. It has so many interesting aspects about it. And so my next question, to get to know more about this research, Daniel, I would like to ask you, why did you decide to join this particular field at this lab? And how did it align to your academic interests?
Yeah, good question. So I actually emailed one of my other supervisors, Dr. Ravi, straight off the bat. And I did it because I saw a journal article out sitting on the web from 2020, must have been 2022, 2021, talking about making these large cell scaffolds for cultivated meat. And I said, oh, that's really interesting, So I literally just went and wrote up an email one day while I was not working at my current job and just sent it off to them, and that's when they interviewed me, and then after a bit they got their funding and
It worked out.
it did.
That's so
That
cool.
is amazing to hear. And what is it about this field that sparks your interest?
What I really like is that this is going in a whole new direction for cultivating, like making products. Ultimately, with making any sort of therapeutic that requires a lot of cells, you need to create a lot. And a lot of our current processes for cell growth are very restricted on what we can do. So especially the aspect looking at bioreactor growth is very critical towards developing not just for, say, meat, but also eventually for the growth of building entire organs and tissues that we may need for humans in medicine.
That is so interesting. Cell culture is quite challenging. It has so many different applications. I've worked in a lab myself, and it's incredible that you're working with whole bioreactors. It could change everything we know about this industries.
So I think a lot of people are like unsure about what materials engineering or alternative proteins, or it commonly comes with the tag lab-grown meat, entails. So could you tell us about what people might misunderstand about alternative proteins?
Yeah. So I hope people are familiar with alternative protein to some extent, but briefly, alternative protein is just a way to make the proteins in unconventional ways. Typically, we are used to have protein from livestock or animal agriculture, right? But there are nowadays a lot of different ways by which protein can be synthesized. I mean, fermentation, bacteria, algae. Cultivated meat is one of the way by which we can produce the animal protein. And there are a lot of misconceptions and misunderstandings that you have asked. And we deal with this because, as I mentioned, under the project, we have different institutions, and there are many researchers who are trying to understand what are these misconceptions, what are the people's perceptions of the cultivated meat. One of the very common misconception is that it's very unnatural, and which is very obvious for people to assume that it's very unnatural. But more from academic point of view, if we get a chance to explain it to people, I mean, we would say it's not very unnatural. It's very much biological, relevant, and similar to the actual meat that we consume or we buy from a grocery store, right? But the argument or the misconception is very much more philosophical, because meat or food is very much tied to some extent culture, tradition of a lot of people and communities. So it's hard to disentangle the biological and philosophical perspective. But I think people are working on languages, on various phrases, how to bridge that gap. But this is a very fundamental misunderstanding, particularly for the cultivated meat industry. Another misconception is that it's a very ultra processed and very unhealthy food. Definitely, there are a lot of unhealthy food in the market, which we all consume to some extent moderately or not. But this is a very another misconception which we also hear from many researchers. For example, we all love chicken nuggets, right? However, if you look at it, only 40 to 50% of the chicken nugget is pure chicken meat.
Wow.
The remaining are basically different ingredients added to increase the flavor or texture or improve the property of the nuggets so that people will consume. In the same way, when we talk about ultra processed food, it's not the processing that can change the health outcome or make it unhealthy, but rather the different ingredients we add to the material. So we don't know, we're not at a stage where we can say whether each and every alternative protein or cultivated meat is going to be ultra processed. There will be many processing stages, definitely, because we are growing the lab. It's not a straight cut from the animal, right? There are going to be several stages, maybe 5 or 10, but each stage does not necessarily going to add any poor outcome to the meat per se. So what we need to understand as it evolves is that what are we adding at each processing stage? Are those unhealthy or not? And the last, one of the last many, I think, misunderstanding is that whether the area of cultivated meat is going to replace the entire farmer. this scope, right? That, I mean, I went to a couple of conferences, and that has been one of the main challenges, that for people to understand how this industry is going to change the existing industry. But again, I mean, as we learn more about this cultivated meat, the way it started evolving, it's not to remove the existence of farmer, but rather to support the increasing demand of protein in the world, in the globe, because of the increasing population. And I see that, I mean, farmers should not consider this as a threat, but rather as an alternative way to support their industry and to meet the demand which they cannot meet given the current infrastructure. So these are the very common misconceptions exist around the area of alternative protein or cultivated meat. I mean, I mentioned cultivated meat, but for example, as I mentioned, there are many ways by which you can make these alternative proteins, right? And people, a lot of times, I mean, even my family members think that, okay, alternative protein means all proteins are same. But that's not the case. I mean, there's tons of alternative proteins, and each protein has its own way of manufacturing, digestion, the bioavailability, the different things that each protein comes with, the pros and cons. So all alternative proteins are not the same. We need to look at each alternative protein from their fundamental building blocks to how they are made, to how they're consumed, and what happens to it in the body after the fact. So there are a lot of differences between the proteins, and I think we should look at each individually at a much more better depth to understand them.
Okay, thank you so much for the insight, because I myself also had a lot of like misconceptions before I learned about what alternative proteins are. And I think a lot of people have questions regarding like, the carbon footprint of animals and how material science can turn sustainability in a different direction. And looking ahead, where do you think alternative proteins will take us in the next direction in research with the next 10, 20 years?
That's okay. That's a very visionary question. But we have seen over the last, say, decade, there has been a significant growth in the area of alternative proteins. Look at the number of companies within Canada. There has been a significant growth in number of companies producing these alternative proteins in different sectors. So definitely, I mean, over the next 10 to 20 years, I expect a lot of improvements and advancements in different stages of particularly manufacturing and scale up. Because one of the main challenge right now in the area of alternative protein is the scale up and manufacturing. A lot of people can make these alternative proteins in a small scale, small batches, wonderful, fine. But the moment they go to scale up in large bioreactors, as I mentioned, these brewery tanks, it fails. So scaling up is a big challenge in the industry. So I hope and I look forward to see that those challenges are being addressed. To add to that, in the future of alternative protein again is, so right now we have different alternative proteins in silos. For example, say different proteins in dairy proteins, in animal proteins, fungal protein. And recently, I was watching a video of a fine dining restaurant in Copenhagen, and where they actually do a lot of fusion food, right? So what I anticipate is that in the future, in the next 10 to 20 years, there will be a lot of amalgamation and fusion of these multitude of alternative proteins to create hybrid products that we don't imagine. So right now we buy one particular protein source from the market we consume, right? But in the future, I see that there will be a lot of integration and amalgamation of this multitude of proteins that will create novel food components, which we could now imagine, but it will be a reality in the future that we can buy and experience those kind of different hybrid products and foods. And lastly, another big challenge is the regulation and standard. So I hope, I mean, we have made a lot of progress over the last few years in terms of standardizing and having a good regulatory workflow. But I think in the next 10, 20 years, we will have much more harmonization globally in the area of alternative proteins, because this is not only pertaining to Canada. It's a global problem of meeting the increasing demand of alternative proteins. So I think there are a lot of efforts going on across the globe. There are organizations who are sitting together every year, to understand how can we streamline, how can we ease the process of regulation, how can we ease the process of standardization. For example, if you have a company tomorrow in the area of alternative protein, You don't want to wait for 2 years to get approval, right? So that timeline of getting approval and getting regulatory approval will be cut down into few months, hopefully. And so that is something which I hope will advance significantly. That will allow more company to work at a much faster pace than what they're doing right now.
Okay, that's very cool, and that's good to know. I think a lot of students are curious, like if they're wanting to work in this field, they want to know like what kind of skills they should develop or something like that. Could you tell us about that insight?
Yeah. So, as I mentioned that again, I'm referring to the initial phase of multi-researcher, multi-institution, right? So so the area of alternative protein is a very interdisciplinary field, which means that people from different backgrounds, be it say mechanical engineering, material science, biomedical engineering, or even social studies, philosophy, they all can contribute in some respect to one another. In terms of skill set, I mean, if the audience is undergraduate students, for example, I don't expect them to have a plethora of skill set to start working in the field of alternative protein, because I mean, they're doing the courseworks and stuff, right? So we don't expect too much of skill set and additional benefits. But the important thing is that you need to have strong fundamentals in your own domain. So, for example, in mechanical engineering, we need to know how this cultivated meat that is being fabricated or any alternative protein finished product is mimicking the real protein structure, the macro structure. So for that, you need to understand the structural properties, right? If somebody is in material science, you need to understand how the surface science, interface science, how the property relationships changes at different processing stages, right? In biomedical engineering, you need to know cell culture, those kind of things, right? So it's not that you have to have a very particular skill set, but having a good fundamental knowledge of your domain is very critical. And then if you can find somebody working in this field in your university, then that's fantastic. Reach out to them. We do have a couple of, always in every summer we have undergraduate students working in the lab. We don't look at particular skill set that they have, but we look at their grades, we look at their backgrounds, how they're strong in the fundamentals, and are they eager to learn? Because this is a new field. We don't expect them that there is a training program that you can go and learn all the skills immediately, right? But if they're eager to learn, then they can certainly join in the summer, and they can at least learn in the next few months the different skill sets that we are using, and that can be a building of the skills rather than having the skills beforehand. Because I don't expect that you have too much time even in undergrad studies to go to the labs and do the research, right? But summer is the particular time. So I think eagerness to learn, having strong fundamentals is very critical in any domain you are in. And as I mentioned, name me a domain, I can tell you how you will contribute to the area of cultivated meat.
Okay. That's really informative. I think it's like really good to know that it's an interdisciplinary field where a lot of students from different backgrounds can contribute.
And it also opens up so many possibilities because, yes, we're in the faculty of science. Hey, sometimes you see people in life sciences or like in biology who think, oh, I only have to do biology or go into health or similar fields. But there's so many interdisciplinary fields where you can also learn so much from other professionals and from other, researchers. And so our next few questions are a little bit more tailored toward those undergraduate students who want to join an interdisciplinary lab, and who are eager to learn. So Daniel, you mentioned that once you were that student, right, you just shot that email, and I know you're here. And so now, if you were to get an undergraduate student to, say, volunteer at the lab and to help you out with a project as a PhD, what would you look for in this undergrad student?
Dr. Sahu actually put it perfectly in that you need to be willing to learn. There's a lot of different topics here that we go through, and it's a lot to take in. But if you want to be in this field, you have to be willing to go through. And even if you don't know everything, because I don't expect them to know everything about every single cell assay we do or step, just being willing to learn, to take a look at the research of what is this doing, why is it doing this, and then obviously ask questions. Like, we're 100% willing to answer why we've chosen these metrics. Like, we put a lot of work and time into this, but wanting to be there and put that time and effort in is very critical, because some days you are in lab for a long period of time and there's nothing you can change so far to do that, to shorten that. So you have to want to be there. Otherwise, you're not going to want to come back the next day.
Of course, and it involves, yes, a lot of say independent research, like going through the literature on your own, really putting the effort to understand, but also, as you mentioned, like asking your peers, reaching out to your mentors. That could be super important in a lab setting too. And now finally, what is the most transferable skill you have learned from your undergraduate and now in your graduate student studies?
So this one's actually going to be an interesting one because it actually does cover everybody. Documentation. Write your notes out. If you are taking good notes through your lab books, into your in all your background supplementary information, you are going to do very well because there's just so much to cover, and having to memorize and remember everything. I don't remember what I did the other day. I have to go pull open my lab notebook. I have to take notes. I have to make little bookmarks to try and pull through. But if you have good documentation skills, you can pull that information up constantly, refer back to it, and then you have a good foothold to essentially spend more of your time thinking about what you need to move forward, what are the problems, and to bootstrap your solutions forward. Otherwise, you're going to forget and end up repeating the same experiment over and over again.
That is so true, and that's such a unique advice. I've never heard that before, and it's very practical too, because you're physically having a record of what you're doing in the lab day in, day out, and then that can turn into like a very valuable record for your other experiences.
Mm-hmm.
So thank you for sharing.
No problem.
And so I believe that unfortunately that has brought us to the very end of our questions. Thank you so much for sharing with us today. We've learned so much from this conversation, and we hope our audience has too. I believe there's probably many students out there who are now interested in alternative protein and also in, enhancing their skills from such an interdisciplinary field.
Definitely.
Yeah, for sure. And this is all for today's episode. We do have a couple initiatives to share.
I know a lot of students are curious about research opportunities, and so I think tuning into the OUR Office of Undergraduate Research, as well as some initiatives by SynBio at McMaster and Alternative Proteins Group at McMaster, they're doing a lot of initiatives around this field. If you're interested, please tune in.
So yeah, symposiums, conferences, and also feel free to give us a follow at Instagram at McMasterOUR and keep up with our newsletter and our websites to get all the newest, most updated information. In our next episode, we will talk about AI and antibiotic discovery with PhD students from Dr. Stokes' lab, so that's pretty exciting as well. Make sure you tune in. But as for now, thank you so much for being here, and I hope you have enjoyed the episode.
Thank you.
Thank you
Thank
so
you for having
much.
me.